Hey, yo, is this thing on? All right, this goes out to my homies out here trying to learn, like, why do we sleep, okay? So sleep, very important, super good for you. Everybody knows you feel better when you sleep, but what's actually happening in your cells? What's going on inside of your brain? Okay, so we got a couple of different things. When you're sleeping, you're not eating. That's called fasting, right? So if you extend that fasting window, like not eating right before bed and, not having, breakfast the first thing when you wake up, then that sleep is going to result in something called autophagy. Autophagy literally means self-eating. Auto means self, phagy means eating. And that is your old busted cells. Your body's like, listen, we are low on food. We are fasting here. We are low on nutrients. We don't have our chomps, our proteins, carbs, nucleic acids, and fats. We don't have it coming in. So we just need to clean things up. Okay, it's time to take a break. Okay, like a fall break, spring cleaning. We take a break. We check all of the resources that we have from the real food that we are eating. That's why it's important to eat living things, because you're a living thing and you want to make cells just like other living things, right? So eat like real stuff that actually grows. That's where you get your proteins, carbs, nucleic acids, lipids, and fats, so that you can build new cells instead of just relying on your old busted cells. When you eat all the time, your body like never has that time to take a break, never has that time to rest. But when you sleep and you stop eating, your body can go through this autophagy cell eating process to go through those old busted cells. Now, if you don't go through those old busted cells, you're more likely to get cancer, Alzheimer's, buildup of proteins and non-functional proteins, non-functional cells inside of the brain. That's all Alzheimer's is. A buildup, cancer is just those old busted cells. They're not really working. They're just going through mitosis over and over and over, just making a whole bunch of cells that aren't really doing something. It's a malfunction because they're not functioning properly, okay? So if you are sleeping well and you're going through autophagy, you're going to hit some mitosis, make some new cells. That's called the cell cycle, cell division, making new cells. It's kind of all the same thing. Now, technically, mitosis is just a part of the cell cycle, and we will talk about that in a second, okay? Now, the other thing that's happening during sleep is cell development. So we have cell growth and we have cell development. If we want to separate those things, we say cell growth is just making a new cell, but then cell development is when it gets good. It's specializing. So your brain cells are going to grow and make connections. That's learning and memory, coordinated by the hippocampus, a part of your brain, where it is taking the information from the day and is reworking it. And that's with your dreams, or just like weird combos of the stuff that you learned from the day, where you're deciding, is this cool? Is this something that goes together? Like, should I keep this in my long-term memory? Like, this is important. So of course, you're going to learn this information better by tomorrow if you take notes on it and then you actually get a good night's sleep. Your brain's like, oh, okay, all right. So it's going to put it together. Now that growth, that's when your neurons are going to reach out and they're going to make new connections with synapses with other neurons, okay? And now that is going to be more solidified. The more you repeat things, the more you learn things, and the more number of times you watch this video and take notes on it, that coordination is going to occur with the hippocampus, and it's just going to solidify that connection. Now that's the development, okay? Your neurons, you're not getting more neurons, you're not getting more brain cells. They are just reaching out, they're developing, they're specializing, they're making those connections, just like trees that grow, okay? We don't want to plant a new tree. That's going to take forever, okay? Well, I don't want new students in my class. I want the students that I have to get better, to develop, right? Which is what we do with doctors. Doctors specialize. They get really good at something, okay? Or like when you are growing up in school and like picking out what you're interested in, right? Like you don't want to go back to kindergarten and start over, right? Like we are moving forward on this. We will need you to develop those cells. Now, that's for like really important cells. That's muscles and brain cells. Those cells are what's said to be in G0. Those G0 cells are not going through the cell cycle. Okay? They are developing, they're specializing, they're getting better, they're older, okay? Now, I'm not talking about old busted, I'm talking like old and better, like good, fully functioning. And there's other cells that are gonna help clean that up. Now, part of the autophagy process could be mitophagy or other cell cleanup. We gotta repair, we gotta replace, we gotta keep those cells that are in G0, we gotta keep them functioning at top notch, okay? So sometimes we don't need to break down the entire cell or eat the whole cell. We can just break down one of the mitochondria that's not functioning very well. That mitophagy, making new mitochondria, that's basically a bacteria that's living inside of each one of your cells, like a bunch of bacteria living inside of each one of your cells. That helps make you ATP, right? Energy to function. It's not making good energy, right? We're going to want to break that down. So that's something that we get from fasting, okay? Clean up the old proteins that aren't working well, clean up the old mitochondria that aren't working well, clean up the old busted cells that aren't working well. If the whole cell just like, all right, it's time to replace this, we're going to recycle it, break it down into its individual pieces. We don't throw it away, really. All those pieces, the proteins, carbs, nucleic acids, lipids, and fats, we can reuse that to make new membranes, to make new nuclei, to make new proteins, new ribosomes, new mitochondria, okay? So that process, mitosis, okay, it takes about 1 day in a eukaryotic cell, like an animal, okay? And it starts with interphase. That interphase is preparing. This is a preparation for mitosis, okay? And preparing is divided into 3 stages. It takes about 23 hours for all of interphase, G1S, G2. Now, G stands for gap. Really, I would like to call it growth, okay? The cell needs to get a little bit bigger because it's preparing to do this splitting cytokinesis. So the cell's getting bigger. It's kind of like duplicating some stuff, just like getting ready. Now, the main thing that's happening in interphase that's cool is in the S phase. S stands for synthesis. And synthesis, it means making. Now, what are we really making in S phase? That takes 8 hours. We are duplicating the DNA, okay? So that S phase there, that is for DNA replication. That's the vocab we need. DNA replication, that occurs in S phase. In preparation for mitosis. Now, interphase taking 23 hours approximately, mitosis taking 1 hour approximately, depends on the cell, depends on a variety of things, okay? That process, interphase gets us ready for mitosis, and then mitosis strictly is just moving the DNA. So it can happen really fast because we spend a lot of time preparing, replicating our DNA, like all 23 pairs of chromosomes, 46 total, like all of the genes that we have there, like the number of proteins we're making is something like that we can make, it's like 10,000, 20,000, something like that. Like there's a lot, okay? There's a lot, 3 billion base pairs. There's a lot of DNA. That's why it takes 8 hours to replicate. And it goes fast, okay, considering how much stuff it is, right? I think I was looking it up that like if you took one tiny little cell and in the nucleus of that cell, you got a full set of DNA, that's called diploid. And that full set of DNA, if you were to take out those 46 chromosomes and you were to string them out, right? Like take this chromatin right there and you were to string it out and stretch it out as far as it goes, we're talking like it would actually be like this long. From one tiny microscopic cell, you spread that out, that's actually like, that's just a massive amount of DNA, which is why when you look at your cells under a microscope, that nucleus is really dark because it's packed with all that freaking DNA. All that DNA, instructions to make proteins, that's what's, you need to have in each cell. So if we're going to take one cell and we're going to split it in 2, now, what happened to the old busted cell? We just told it to break itself down. Now we got materials to make a new cell. But then we have a nice young fresh cell say, hey, I'm going to replace that other cell by going through the cell cycle. It's interphase, mitosis, cytokinesis, and then we make a new cell, we replace that, okay? But we have to duplicate the DNA first, okay? And we're going to duplicate that DNA when it's in chromatin form. It's just like hanging out inside of the nucleus. And this looks messy, but this is accessible, okay? Like your room, your house, things might be messy, but you know where things are and you can find them, okay? So we have this chromatin form all throughout interphase. It's in chromatin form, okay? And we're going to duplicate it. And so there will be double the DNA. You can't really tell because it's just all junked up in there, okay? It's all just in chromatin form. Unpacked, easy to read, transcribe, easy to make proteins, okay? That's the open. Then we get into mitosis. That's business time. That's where we are moving the DNA. We have to split it perfectly in half to make sure that there is a full diploid set on one side, full diploid set on the other side, and then we can spread, pinch, pop, okay, for cytokinesis. So when we think about mitosis, think about moving DNA. Now, is DNA just going to like magically move, magically pack itself up into chromosomes? No. Okay, so we're going to have to move it around. So we're going to have some proteins, because proteins do work. Enzymes are the proteins that do work, powered by ATP, thanks mitochondria. We're going to have to line them up in the middle, and then split them, and then we can split the entire cell, okay? So this is done in 4 phases, which I like to remember as PMAT. Prophase, metaphase, anaphase, telophase, okay? Pro means first, meta means middle, ana means apart, and telo means end. One way to remember it, And phase just means a period of time. So in prophase, what we have here is that we have a nucleus around our DNA in chromatin form. But if we're going to move the DNA, we're going to want to pack this up into chromosome form. And that chromosome is a lot easier to use. Of course, moving anything, pack it up, it's going to be a lot easier there, okay? And so we are going to pack it up. I like to draw the chromatin going to chromosomes right there and label it chromatin packed into chromosomes, okay? Then you also show a dotted line for the nucleus because it's being disassembled. Now, all nuclei and cell membranes is all made of phospholipids, okay, in a bilayer. That phospholipid bilayer, that's every membrane. So we're just going to break that down so that we can get these things and spread them out in the middle of the cell, okay? Now, again, spread them out. We got centrioles, spindle fibers. Those are going to be built here in prophase. Prophase is a busy time. That's like the biggest of all these four. Prophase is the biggest one, okay? It takes a lot of time. You are building those centrioles and spindle fibers, okay? You are breaking down that nucleus, packing up the chromosomes. Then there's this tug of war process that happens. They all reach in and attach there, and then this tug of war process lines them up in the middle for metaphase. Now that middle for metaphase right there, okay, that's when the cell just says, hey, let's check real quick. Are we lined up in the middle? All right. Now the cool thing about this tug of war is everybody wins, okay? In this spot, you see that we have separated the X's, and the half of them, like when you separate the X, it's going to look like this, right? And pull, and we're dragging them through, and they look like that, all right? Now this one's not packed very well. We need to pack up that chromosome a little bit better, but they together like that, and then pulled apart by the centrioles and spindle fibers, okay? So, and that's what we see in anaphase, are being pulled apart, right? Now, how does it do that? The centrioles are going to literally eat the spindle fibers. They're going to like break them down. They're just like eating, and that shortens them, and then boom, both sides win. So we have then a full set of DNA over here, a full set of DNA over there. That is what we call diploid, okay? Whereas at the beginning, we was quadruploid, meaning that we got four of each chromosome, because at the end, we want this to be a diploid cell. Diploid means two, because of course you have two parents. So when you were made, all your cells are supposed to have half from your mom, half from your dad. Half from mom came from the egg, that's haploid. Half from dad came from the sperm, that's another haploid. You put that together, boom, fertilization, zygote. And then you have a diploid cell. So what you want to do is take an original diploid cell, replicate the DNA in S phase, then you're going to have quadruploid, then you split it, diploid, boom, two diploid cells, okay? And then they can start developing and becoming their own thing. Technically, cytokinesis is after mitosis, okay? It's a pretty short process there, a couple of minutes maybe, okay? But let's make sure we know what happens in telophase. It's like the reverse of prophase. In telophase, we don't need our centrioles and spindle fibers anymore, so we can start breaking those down. We don't need them anymore, okay? Break those proteins down. Then the DNA, we're going to wrap it back up, reassemble two nuclei now, so the DNA can be safe. And then, once it's safe, we're done. We've got this all packed up, so we can take that and unpack it back into chromatin, so it's nice and easy to use, okay? So we see that unpacking back into, unpack into, I wrote this wrong. Unpack into chromatin from chromosomes, okay? It's unpacked because it's nice and safe there as we rebuild the nucleus, reassemble it, but break down the centrioles and spindle fibers there, okay? And then what we are seeing there is the nuclei with the chromatin DNA in there, okay? Then it's going to split right there. It was coming from there, so we see from this, transition to this. I mean, I know it's smaller there, but it's the same thing. Okay, we've got the DNA in chromatin form, boom, okay? All right, and that's that process that can happen if you eat right, you sleep right, okay? And you got the materials to make a new cell, and you're giving your cell that time to sleep, take a break, and give yourself those fresh new cells, right? Okay, that's good.